This method can help answer key questions in the environmental microbiology field about the ubiquity of microbes capable of EET, especially in low biomass or geochemically extreme conditions. The main advantage of our on-site microbial enrichment is that it doesn't require a power source, facilitating a month-long isolation period for the capture of difficult-to-culture taxa.
Generally speaking, individuals new to this method will struggle because electrochemical system fabrication contains multiple steps that require a precise implementation.
To construct the fuel cell Type II electrode incubation system, twist an insulated wire of the appropriate experimental length together with a lead of titanium wire from the electrodes and cover the connections with water-resistant wax. Then, further protect the cables with marine-grade heat-shrink tubes and connect the two wires with a cathode and an anode by a resistor of known resistance. To measure the voltage temperature logging over time, check the voltage between the ends of the resistor to allow estimation of the current production from the fuel cell reaction. And use a data logging voltmeter with the appropriate connections leading to the anode and cathode to measure the voltage difference over time according to the manufacturer's specifications. To protect the data logger and electric connections, place a plastic bag around the logger and all of the electric connections, and secure the bag and cables tightly to protect the materials from a strong wind. To collect an electrode sample from the natural environment, at least 30 minutes prior to collection,